Hello, welcome back. So as we move through chapter 11, I'm kind of rearranging things a little bit. It seems to make a little bit more sense going forward this way. But this little mini lesson is going to talk about different replication processes that prokaryotic cells use, and then just a quick mention of endospores, which we've already covered, so I'm not going to spend too much time on that. When a bacteria replicate, a typical bacterium, or I guess I should say archaea as well, so let me use the word prokaryote. When a typical prokaryote replicates, it uses this process of what's called binary fission. To compare that to when a eukaryotic cell replicates, it's called meiosis or mitosis, right? So mitosis, eukaryotic cell, nuclear division, cytokinesis, all that kind of stuff. Bacteria, archaea, these prokaryotic cells do not have to do that because they don't have a nucleus with 46 chromosomes and all of the organelles that needs to be divided. They basically just have their one circular chromosome. So binary fission is quick and easy and pretty straightforward. So let's go ahead and go through that. So a bacterium has a single circular chromosome. That's its genetic material. When it's getting ready to divide, it will just copy. And so now we have two copies of the circular chromosome. They're kind of the light and the darker, or the bluish purple and the pinkish purple shades there. So you have your two copies of genetic information. And the DNA replication is exactly the same. So your leading strand, lagging strand, Okazaki fragments, helicase, unwinding, DNA polymerase, all of that is done exactly the same in a prokaryotic cell and a eukaryotic cell. So that's all the same. The cell grows a little bit bigger. So step 2, the cytoplasmic membrane elongates, the DNA molecules kind of get further apart, and eventually you start to kind of pinch the cell in half. So the cell membrane starts to also pinch, the cell wall starts to pinch, and eventually you get a completely cross wall forms. The two chromosomes are separated from each other and your daughter cells separate. Now, in an organism that has particular arrangements, they may not fully separate like that. They may stay stuck together, and that's what leads to your diploid and strepto and staphylo and tetrad and V-shape and all those types of things. So here over on the electron micrograph, here are two bacilli that probably just got done finishing up their binary fission, and they're still stuck together right there at that cross wall. And here's a couple other ones over here that are kind of in that same time frame. So looking at that particular picture, it would be hard to determine whether this organism tends to be single bacillus or diplobacillus because it's about half, maybe a little bit more single, but there's quite a few diplo here. So you'd have to see a lot more samples to determine if this particular species likes the arrangement of diplo, or we're just happening to see them before they completely separate at the end of their binary fission. So it's hard to tell sometimes. In a slightly different version of binary fission, there is a form called snapping division where it follows all of the same steps, but our particular characteristic of the bacteria that can do this is they have built up kind of pressure that once they, here's this outer portion of the cell wall, and then once that outer portion ruptures, it kind of goes from this to this. It kind of does a quick snap. And so we can see that here would be kind of the very beginning. It looks like we have the division, right? The outer cell wall, and then we get this quick snap. And so now we have it in this V shape. And then it can continue to do that and stay in that V shape, or it might lead to the palisades where they're kind of stacked side by side to each other. So snapping division is just a unique form of binary fission that allows for that V shape to form in the arrangement. Some bacteria don't do binary fission or snapping division. They produce spores, reproductive spores. And these are typical for bacteria that are filamentous. So instead of being a bacillus or a coccus or a sporillum or a spirochete, we didn't cover this, but there are some bacteria that are long and stringy, like a spiral, almost look like a curly fry. So they don't go through binary fission the normal way. They will replicate their DNA, but instead of doing an equal split, because that would be hard to do with the long filamentous bacteria, they produce a little reproductive spore at the end of their filament. And so here's like another one right here. There's another little one right here. So they send their DNA, they kind of pinch off this little spore. That spore will go and grow into a new long filamentous bacteria. And the group of bacteria that are commonly replicate this way are called actinomycetes. They're usually living in the soil. They're long and filamentous. Another form of replication for certain microbes is called budding. It's kind of like binary fission, but instead of splitting off equally, a smaller little bud kind of gets pinched off. So here's one right here. Yeast do that. So your Saccharomyces cerevisiae, it basically is just baker's yeast. They replicate via budding, the budding process. So they're a eukaryotic cell, so they're not going to do binary fission anyways, but some bacteria can also do budding as well. And then lastly, this chapter mentions endospores. We already talked about the process of sporulation. So the Bacillus and Clostridium genus can produce endospores. Again, they're resistant to most types of disinfectants, drying, heat, radiation, lethal chemicals, and some of these genus can produce pretty nasty diseases like anthrax, botulism, tetanus, Clostridium difficile, right? That really bad diarrhea that can pass through healthcare facilities, those types of things. Okay, so I think that's it for this section. I will see you next time. Bye.